हाँ.
Done.
Okay, thank you. Good evening to one and all present for today's session. I am Megha Hundekar, research scholar from Mysore University. And First, to I would like to introduce everyone today to our Dr. Madonna ma'am is a passionate biotechnologist who has over 8 years of experience in research and even has been working in both government funded projects and the industry. Ma'am has completed her academics from MTech from Shastra University, India, and she has completed her doctorate in nanobiotechnology from VIT University, Vellore. Ma'am presently is working as a director in Zygene Biotechnology in the research and development sector. And ma'am has overall almost 10 international publications and is also awarded with few awards like she has been the subject topper in 2008. She has been awarded for her publication at VIT University, India. And she has also received Senior Research Fellowship from CSIR. So today we are very happy to have Dr. Medona ma'am for today's session. And I expect today's participant will be very happy to get familiar with what is the organization and what are the provisions available at their organization. So I would like to request Dr. Medona ma'am to take over the session. Over to you, ma'am.
Thank you, Megha. Good evening, Dr. Arvind, Megha, organizers, and every distinguished guest who are present here today. My name is Madonna, and currently I'm working as a director at Zygene Biotechnologies Private Limited. Today is a Monday evening, after long day. I think some of us will be very tired. I'll try to keep my presentation as short as possible. Today's presentation basically, through this presentation, I wanted to introduce our company, and I wanted you to know how our industry works, what are happening in my company. Also, I wanted to find out from your side, as in what challenges you're facing in your research, so that we can also get an idea of what further our product portfolios has to improve. So I hope it will be a useful session for both of us. Shall I start sharing?
Yes.
Sure, ma'am. Please sharing your screen.
Okay. Okay. So our company is a basically molecular diagnostic company founded in 2019, and it is placed in a sprawling campus of biotechnology and industrial zone in Kinfra High Tech Park, which is situated in Kochi. So we have a department, established department for R&D, production, and bioservices division. And our basic mission is to address a diverse range of challenges, whatever challenges that is coming in molecular diagnostics market. So throughout our process, we have also involved in other works that are related to bioscience, not just molecular diagnostics, although our product portfolio basically go around molecular diagnostics, and it is our final vision. But still, we also work with other biofields like phytochemistry, microbiology, cell culture, and so on, genomics, and so on. So our basic portfolio is, at present, we are working on nucleic acid extraction kits, qPCR diagnostic kits, DNA barcoding kits, and biochemical assay kits. Nucleic acid extraction kits are available in the market in under the trade names Zymac TM and Zypure TM. Zymac TM is actually basically magnetic bead based nucleic acid extraction kit, while Zypure is conventional column based kits. And we are working on, at present we have some approval going on, so we are working on our qPSA diagnostic kits, and soon DNA barcoding kits and biochemical assay kits, basically antioxidant assay kits, will come to market in most probably in this month. So one of our main product is Zymag. Okay, Zymag, you might be familiar with all sort of nucleic acid extraction kits that are available in the market. Still, I wanted to introduce or I wanted to let you know about the different types of nucleic acid extraction methods that are available, that are currently being in use. Generally, conventionally, we can use phenol chloroform isolation method, which takes about 1 to 2 days, while it is much easier when it comes to column-based nucleic acid extraction method. Why Zymac is actually magnetic bead based extraction technology. It actually provides instrument-free processing. So instead of using a column, we can make use of magnetic bead for better extraction of DNA and RNA. This technology is actually very high throughput friendly. That means it can be used in automated systems. The importance of Zymac is that it is actually simple. Its extraction protocol is very simple and it can be finished in less than 30 minutes, unlike other kits. We can actually get very large amount of high yield from very small amount of sample that we are starting with. If we have a very small amount of starting material, we can still get a very good yield of DNA through this magnetic bead extraction method, because it has a strong buffer system that makes all these downstream processing easier. So just wanted to introduce to those who doesn't know how a magnetic bead extraction kit works. It has got four basic steps. First is lysis, binding, washing, elution. Let us take any sample, let it be tissue, animal tissue, or fish tail, saliva, soil, whatever it is, the initial sample, we have to just lyse it using RNase or proteinase kit, depends on the whatever protocol as I mentioned in the protocol, and just incubate for 5 minutes in water bath or in dry bath at 56 degrees Celsius, or at the temperature that is mentioned according to the protocol, and then add beads, magnetic beads. That is, here it is mentioned ZMB, Zymac magnetic beads. Upon addition of the beads, the DNA after that is released after the lysis of the cells will go and bind to the specialized functionalized magnetic bead, and this can be bound to the magnetic stand through when the beads are kept on the, or when the vials are kept on the magnetic stand. The magnetic beads will come aggregated to one side and you can remove the supernatant. This step takes about 5 minutes, and later on you can wash these beads through the same process and then elute using elution buffer in the same way. So it just takes around 20 to 30 minutes, 20 for an expert, 30 or 35 minutes, depending on your sample. So we have about 8 different DNA extraction kit under this label. One is blood DNA extraction kit, plant and algal DNA extraction kit, cells and tissue DNA extraction kit, saliva DNA extraction kit, microbial DNA extraction kit, environmental DNA extraction kit, fecal DNA extraction kit, and bacterial RNA extraction kit. These kits form the basis for our high throughput mechanism, where automated nucleic acid extraction systems are working. So for that, these are modified to form different platforms for high throughput extraction of DNA. which under Zymac we have express saliva DNA extraction kit, which is also used for high throughput. Similarly, microbial DNA plant nucleic acid extraction kit, PCR cleanup kit, magnetic bead for. We are also providing magnetic bead as such for extraction and purification, where people, if somebody wants to customize their work using the magnetic bead, they can go for the magnetic bead as such. And fast bind magnetic separation stand. This separation stand is where it is 1.5 ml capacity centrifuge tube capacity vial that can be placed on this for the extraction process. So we have done some verification validation studies, and we found out that we have excellent DNA compared to conventional or whatever available in the market. At least they are par with the international standard ones that are being imported in India. And we get a good CTU values when it comes to RT-PCR, that which shows that our kits are not showing any inhibition for PCR. And we also get a good chromatogram when we go for sequencing and also other downstream process. We have again, we also have conventional or what has been used for last maybe 10-20 years, column based kits, which is again we have blood DNA, plant and algae, cells and tissue, saliva, microbial DNA, environmental DNA, and gel extraction kit. So these are the basic products that are present in market. Apart from that, we are working on, as I've told you before, some IVD kits, qPCR kits, DNA barcoding kits, and our antioxidant kits are going to be launched very soon, probably in this month. So apart from that, we also provided so bioservices in our company. Bioservices division started 4 years before. It started as a supporting system for our product company, product development. But again, we had plans to stop once our products started coming in market. We had planned to stop services, but considering the kind of demand we have, kind of people who are requesting for other services and all that, we have made it into a different service division, and it's working excellently well right now. So we provided some genomic services, product formulation studies, cell culture assays, microbial analysis. In general, we actually take up big projects where we have to do this formulations, wound healing products, some POC works and all that. But we are also taking small works from research scholars who are really in need of it. So we are also doing microbial analysis for these are part of bigger projects that we have done. So we have already done how many customers, like more than maybe 300-350 customers we have already served so far. And we are still going on with our services work. As how in our company we have an in-house R&D laboratory, an advanced laboratory with PCR, RT-PCR, high throughput facilities, everything that is needed for supporting our product development and bioservice work. We have equipments accordingly, and we have a strong quality control and assurance system. We are going by all regulatory compliance. And also it's very important that when it comes to bioservices, we are keeping client privacy as such it's very important to us. So we go for all the regulatory compliances accordingly. And we have a good trained and expert staffs, and we have a good production facility that supports our company. Our core team includes Dr. Rishad KS. He is the founder and CEO, and he has already been working in industry for about 2014 to 16 years. And he has founded this company for initially exclusively for product development, but later on we had taken up bioservices and contract research works also. So he is a founder and CEO, and he has a PhD in microbiology from MG University, Kerala. and myself. And then we have Arjun Asa. He is a senior scientific associate. He has been associated with the company from the inception of the company. And we have collaborators where we work for product collaboration, product development. We work with Malabar Cancer Center, University of Calicut, and CUSAT, which is Cochin University, and also for bioservices and other related works. We have industrial collaboration with Unibiosys and Albio, and these are just the ones that are very closely involved with us. Apart from that, across Kerala and in Hyderabad and in Bangalore, we have supportive systems where we get our in case for our other services work done to get our services work done. So we are basically situated in Kalamasheri, Ernakulam, and this is our telephone number, and if you have any services inquiry, you can just drop in a mail at services@zygene.in. And if you need any technical support, if you wanted to formulate something, or if you wanted to get some idea about the work done or anything, you can just mail in to support@zygene.in. And nevertheless, you can visit us at www.zygene.in. Thank you.
Thank you, ma'am. The session is open for question and answers. Anyone in the participant, if want to ask any question, you can unmute yourself and ask the questions.
May I interrupt?
Yeah, sure, ma'am.
Okay. I also wanted to know what sort of challenge you're facing. So we wanted to actually improve our product portfolio. So we are also thinking of making kits that helps with the microbiology works and all that. If there is anything that can make the work easier for you.
Yeah. Actually, ma'am, there are many problems that we will face, but at present, like might be because the participants are less, it's not possible to have the discussion. Anyone in the participants, if you have any query, you can please unmute yourself. Actually, today, just because it's a working day, I guess there are many participants who have not joined the webinar.
That's fine. We can actually share our flyers and all that. And I would also like to share a questionnaire so that I like to know. I'm honestly wanted to know, wanted to have this discussion because I wanted to know what sort of challenge researchers are facing.
Sure,
So
ma'am. It will be actually helpful if you create a Google form and just forward it in the group if you're there in the group, or you can forward me so that we will circulate in all the groups of microbiology society and at least gather some kind of survey and analyze even for your organization it will be helpful, and even for us being the members of the group it will be helpful for us.
that's good. I'll make the questionnaire and send it over.
Sure, ma'am. Sure, it will be really of great help for you also as well as for our group as well, ma'am.
The interface is quite important because of this.
Yes, ma'am. Yes, very truly said. So I guess there are no questions for today, ma'am. So I would like to end this session. It was really very thankful out of the entire busy schedule you have taken out time for today and given your time in explaining about your organization and what are the products that your company has launched out in the market and being helpful for the research scholars in the entire field of life sciences. So I owe you a sense of gratitude from the entire microbiology society for giving your valuable time and explaining about your organization. Thank you so much, ma'am.
Thank you, Megha. Thank you. It was my pleasure.
Sure. And I would also like to thank Dr. Deshmukh sir, who every day gives us opportunity to get learn new things, meet new people, and understand how exactly is the direction of life science going and where are we lacking behind so that we can upgrade ourselves. So I would also like to thank Dr. Deshmukh sir. And last but not the least, I would also like to thank all the members who have joined for today's webinar, though it was a very busy day, being the first day of the week. So thank you all the participants. Thank you, Madonna ma'am.
Thank
Thanks a
you.
lot.
Thanks
Yeah.
to you and all applause to all your organizational teams, because it's kind of you're doing a commendable job.
Thank you, ma'am. Thanks a lot, ma'am.
Okay.
Thank you.
Okay.
Thank you. Yeah.
Have a good day.
Thank you, ma'am. So should I leave the meeting, ma'am?
Sure, sure. You can leave the meeting.
Thank you, ma'am.